Hello everyone, welcome to Bioexplaining. This is the third part of our topic, sexual reproduction in flowering plants. So let's begin. In the last two videos, that is part 1 and part 2 of the same topic, we have discussed in details about the male reproductive part, the pollen grain, as well as the female reproductive part and the female gamete, that is the egg, right? So now that you have already understood about the male and the female reproductive parts, their gametes that they are producing, the next step is pollination, right? That means the anthers will produce the pollen grains and these pollen grains need to be transported to the stigma. So pollination can be of different types. There are various agents for pollination. It can also be done artificially, which is artificial hybridization. So I have created 4 separate videos based on all of these topics in detail. So I'm giving the link of the playlist here named as Pollination in Plants. I would request all of you to first go and watch those 4 videos in sequential order, so that you have a very clear concept of pollination as a whole. Only then, please come back here and then we can continue with the topic. So what do we have? We have the female reproductive part, that is the pistil, right? So this is the female reproductive part. The top part here, this is the stigma. Then the long slender part, this is the style, and the basal part, the bulged part, this is the ovary. And inside the ovary, what do we have? Inside the ovary, we have the various ovules to be present. Okay. So what has happened after pollination? During pollination, the pollen grains have come and they have landed on the stigma, right? So what is this? This is the pollen grain which has now landed on the stigma. Okay, so the pollen grain now has to enter through the stigma, move through the style, and then enter the ovules, right? So how does this happen? The thing is that the ability of the pistil to recognize the pollen is followed by its acceptance or rejection. And this is the result of a continuous dialogue between the pollen grain and the pistil, right? So what do we mean by it? That the pollen grain is coming and sitting on the stigma. Now the stigma can either accept it or the stigma might not accept it. The stigma might also reject the pollen grain. And this is done on the basis of what? This is on the basis of a continuous dialogue. That say I want to enter somebody's house, so I have to have a dialogue with the security guard there. So similarly, if the pollen grain wants to enter the stigma, it needs to have a dialogue with the stigma, right? Now, what is their dialogue? Their form of dialogue is mediated by chemical components, right? So it is the interaction between various chemicals that are secreted by the pollen grain and those that are secreted by the pistil. Right. So these are clubbed under a common heading that you can call to be the pollen pistil interaction. Okay. So this is the pollen pistil interaction. That is, the pollen grains will interact with the pistil and then the pistil will decide whether it will accept the pollen grain or reject the pollen grain. Right. So these are all what? These are all post-pollination events. Understood properly? Now, what happens is, in this pollen-pistil interaction, if you have the right type of pollen, then the stigma accepts it. What do we mean by the right type of pollen? Right type of pollen means that, say it is of the same species. That is one thing. It can be of the same species, or say it is the compatible one. Compatible. So if it is the right type of pollen, that is it belongs to the same species as the stigma, or it is compatible, then the stigma will accept it. But if it is the wrong pollen, what do I mean when I say no wrong pollen? It might be non-compatible, right? Or say if the pollen belongs to some different species. In that case, what will happen? The stigma will straight off reject the pollen grain. What do we mean when we say the stigma rejects the pollen grain? Rejects means that pollen germination on the stigma will not happen and the pollen tube will not be able to grow through the style. Are we clear that only if we have a compatible pollen grain, same species pollen grain, the stigma is going to accept it. Fine. Now, what happens is the pollen grains have fallen on the stigma. So the stigma has some kind of a sticky secretion. So the pollens are stuck to the stigma due to the sticky secretions. Say this is the stigma region like this. The pollens are coming and they are stuck on the stigma due to some kind of sticky secretion that's there. Okay, next what will happen is the pollen grain will start germinating. And for the pollen to germinate, what are the things that are required? There will be requirement of moisture and nutrients should be available on the stigmatic disc. What do we mean by the stigmatic disc? This part, the receiving part of the stigma where the pollen grain comes and sits, right? So the pollen comes and sits on the stigma. It is stuck due to some kind of secretion from the pistil. And now if proper moisture and correct amount of nutrients are available, the pollen grain will start germinating. Is it good? So what happens is from the pollen grain, a cytoplasmic outgrowth will come out, right? That means say if this is the pollen grain, right? And these are the external coverings we've studied, right? The exine, and this is the intine inside. So what will happen through these gaps, which are the germ pores that are present, a cytoplasmic extension will start coming out. That is, the cytoplasm will extend. So what is the cytoplasmic extension known as? This cytoplasmic extension is known as the pollen tube, right? And this pollen tube will be containing the gametes. Are we good till now? Now, what happens is that after this thing, what will happen? The pollen tube will start growing. While the pollen tube starts growing, it will keep secreting different types of enzymes. What is the function of those enzymes? Because the pollen tube needs to grow through the stigma, right? So it has to dissolve the tissues of the style in order to grow through it. So it will digest the tissues of the style. It will digest the tissues of the style and then the pollen tube will grow. So it grows chemotropically and intercellularly into the style due to a concentration gradient of calcium boron inositol sugar complex. We just said, oh, there's a chemical interaction, a dialogue that happens at the stigma. So this kind of a chemical interaction keeps on happening. That is, in the stigma, there is a concentration gradient of calcium, boron and inositol sugar complex. And due to this, the pollen tube keeps on extending downwards until it reaches the ovule. Getting it? So finally, what will happen? The pollen tube will be entering the embryo sac through one of the synergids. I guess we are clear till now? Well, so now we have understood till a point that the pollen tube has extended through the style and it is entering the embryo sac. So for a very quick recap of what we have studied in our previous video, let us understand what the internal structure of the embryo sac is, right? So what do we have? This side is our micropylar end. Let me consider this to be the micropylar end, and the opposite one to be the chalazal end, right? So what do we have at the micropylar end? Micropylar end has three separate cells. Okay. What are these three cells? Out of these three cells, two cells are known as the synergids, also pronounced as synergid, and the middle one, this one, is the egg cell. And all together, these three, the synergids as well as the egg cell, they constitute the egg apparatus. Okay. At the central part of the cell, or the central part of the embryo sac, we have one cell which is known as the central cell. And this central cell has two nuclei in it, right? So what are these known as? These two nuclei, they are known as the polar nuclei. At the chalazal end, we again have three other cells. So these three cells are named as the antipodal cell, or simply we call them to be the antipodals. Okay, these three are the antipodals. So this is what have I drawn here? This is the embryo sac that I've drawn in a mature state. So this represents the female gametophyte. Now what we see is when the pollen tube enters, the pollen tube, what does it contain? The pollen tube contains two male gametes. Okay? The pollen tube has two male gametes, right? So the pollen tube will be entering via the synergids. Here next to the synergids, you have the filiform apparatus present. So if you remember, we have studied that the role of the filiform apparatus is to guide the pollen tube to enter the embryo sac. Pollen tube has two male gametes. So once it enters the embryo sac via the synergids, it will release the two male gametes one by one. Okay, what happens to the first male gamete? The first male gamete will fuse with the egg cell. Okay, so the first male gamete fuses with the egg cell plus male gamete. And what will this give rise to? This leads to the formation of the zygote. The second male gamete will come and fuse with the central cell. So this is the other male gamete which comes and fuses with the central cell, and this will give rise to the endosperm. Right? So there are two fertilization that is happening. The first fertilization is when a male gamete is interacting or fusing with the egg cell to form the zygote, and the second fertilization is when the other male gamete is fusing with the central cell with the two polar nuclei, leading to the formation of the endosperm. So because two fertilizations are happening, we call this process to be double fertilization, right? So what is double fertilization? Double fertilization is one male gamete will fuse with the egg cell. The male gamete is haploid, that is, it has N ploidy. Egg cell is also haploid, N ploidy. It will lead to the formation of the zygote. So N plus N will give rise to 2N, that is the diploid zygote. So this is known as syngamy. The fusion of the male gamete with the egg cell to form the zygote is known as the syngamy. So for double fertilization, this is fertilization number 1. And what is fertilization number 2? The other male gamete, which is again haploid, N, that will interact with or fuse with the polar cell. The polar cell is N plus N. Why N plus N? Because it has two polar nuclei. The polar cell is the central cell. I hope you're getting it right. The central cell or the polar cell has two polar nuclei which now fuses with the male gamete. And this leads to the formation of the PEN. What is PEN? Primary endosperm nucleus. So this gives rise to a triploid primary endosperm nucleus, right? This is known as triple fusion. Why triple fusion? Because this is one nucleus, this is the other nucleus, this is the third nucleus. So three nuclei fusing, hence this is known as triple fusion, right? So the zygote develops into the embryo while the primary endosperm nucleus is the primary endosperm cell. We are getting it? So with this, we come to the end of the concept of double fertilization. So in the next video, we are going to talk about what happens after fertilization. In this video, we spoke about what happens till double fertilization. Next video, we'll be dealing with the post-fertilization events, what happens to the various structures, etc., etc. Right? So please stay tuned with us. And if you like the video, please drop a like, comment, and share it. Also, please don't forget to subscribe to my channel Bioexplaining. Once again, thank you and let's keep learning. Bye-bye.